Are you ready to embark on a journey of success? To discover your true potential in the untold stories of careers in life sciences? Welcome to PATHFINDER by the Purdue Institute of Inflammation, Immunology and Infectious Disease. With your host, Tommy Soares, discover the untold stories of industry scientists and unveil the secrets to excelling in your career. Thank you for joining us once again, and remember to hit the like button and subscribe to our channel. Welcome, everybody. My name is Tommy Soares. I'm Director of Scientific Strategy and Relations for the Purdue Institute of Inflammation, Immunology and Infectious Disease. And today on PATHFINDER, I have the pleasure of welcoming Ernesto Nakayasu, the Biostructure and Function Team Lead at the Pacific Northwest National Laboratory. Dr. Nakayasu received his degree in biological sciences from the University of Sao Paulo in Brazil and a PhD in pathobiology from the University of Texas at El Paso. After postdoctoral training at Pacific Northwest National Laboratory and a period as a scientist at Purdue University, Ernesto returned to the Pacific Northwest National Lab in 2015, where he is currently a senior scientist and a team leader. His research has focused on the application of advanced mass spectrometry technology, such as proteomics, lipidomics, metabolomics, and analysis of protein post-translational modifications to study complex biological systems and specializing in infectious disease and type 1 diabetes. It is so good to catch up with you again, Ernesto. Thank you so much for being on the program. Thank you for being our Pathfinder today.
Well, it's my pleasure. It's so good to see you again, Tommy.
It's great to see you too. And I'm so excited for the audience to really be able to hear your story, because I think your journey is so unique. And you've gone such a great way. And I think you're probably the first national lab scientist that we've had the opportunity to interview on Pathfinder. And I really want everybody to understand what it's like and what's the difference between, a national lab and a regular academic lab or an industry lab. I know you've traversed all over the world, and I know you're originally from Brazil too. And it's so great to be able to open up the lid and take you back into your past and tell us a little bit how you got to where you are.
Well, I I grew up in a farm back in Brazil, and so I always have like this interest for science, because I was saying, well, so that should be better ways to do things, and better ways to say grow vegetables and like make things more resistant to disease. So that's why I pursued to go to biology and study biology, and then I went to University of Sao Paulo to study it. And at that time, actually, my first thought was to work more on plant, genetic manipulations. But then when I get to my school, was not very strong on that, and I went actually to work with mosquitoes. And to work with mosquitoes, we're studying salivary glands and some antimicrobial factors in salivary glands, and then from there I jumped to working in spiders. So, very big tarantula spiders, and try to understand how their coagulation system works. So when I was finishing undergrad, then I was looking for grad school everywhere in the world. So then actually I started grad school back in the University of Sao Paulo, but as soon after I start, my PhD supervisor got hired at the University of Texas, El Paso. So then he invited me to come. I went to El Paso, which was quite different from Brazil, yes, you can imagine. So before I get there, actually, I went to Scotland for a period of 2 months, actually, for some training and running some samples in a mass spec core facility there. And I actually didn't have much time to really research about El Paso, but So I was thinking that I was going to get like, green Texas with a bunch of cows and the cowboys. But, who knows El Paso? El Paso is far west Texas and it's completely different. So when we were landing the airplane, I said, wait, are we landing on the moon somewhere? So that's the way I got to US, and then from there it's just history, right? So I did my PhD working with Trypanosoma cruzi, which is a parasite of Chagas disease, which is more endemic in Central and South America, but there are quite a bit cases the southern US, from say California all the way to Florida. And then from the Chagas disease stuff, then I came to Pacific Northwest, to to work more on proteomics, and this time of bacterial infections, more specific to Salmonella. And I did also some work also in Legionella and Francisella. They're three different pathogenic bacteria. From there, and I had the pleasure to come to Purdue and work with you, actually, which was a very good time. It was a time that I learned a lot, and I met a lot of very good scientists there. And then there was opportunity to come back to Pacific Northwest, and in 2015. And actually, at that time was to start work on diabetes, which was something that was completely new for me. But I think that is like starting from a little bit about the national lab. That is something that the national lab allows, is for you to pursue some new pathways, right? So you carve your own pathway here. It's not like most in academia that you hired because your expertise in that small field. So you hire, but you have the expectation that you might to contribute to other projects too. So that's how I end up coming here, end up working type 1 diabetes, which I had really no background at that time. And then from there it was, it has been a good journey to say,
Awesome. I can imagine what a journey, because you put it in such a simple way, but my gosh, a jump from Sao Paulo, Brazil to Scotland. Where in Scotland?
It was
Glasgow?
in Dundee.
In Dundee. Okay. So I jumped to Scotland from Brazil for a couple of months stint to learn techniques, and then straight to El Paso. You didn't even get to go back to Brazil. You went straight.
I did. I actually spent 2 days in Brazil.
Enough
Just
to
the New
do,
Year.
yes, enough to
It was
do
I
laundry.
flew back on the 31st there, and I took off on the 2nd.
Wow. Enough to do laundry and then pack the bags and go again.
Yes.
Tell us a little bit about what was it like in Sao Paulo? I mean, you talk about spiders. What was the interest in coagulation factors coming from tarantulas? What was it more like the experience of working with particular scientists that you were exposed to there?
No, I mean, the interest at that point was to understand, because insects are very important vectors, not just for human disease, but for plants now too. And they understand their immune response. So the coagulation is part of the immune response too, especially for insects, so it's a good chunk of their immune response. So understand their immune response may be that we can, block some transmission of disease.
Very interesting. And so then the prof that you were working with got this position in El Paso.
It was not the same professor. So when I start working with a professor called his name is Osvaldo Marinotti, but I was working with this mosquito project, and then he left. So then I went to work the other professor in the lab. Her name is Sirlei Dafre. She just retired a couple years ago, and she was working on spiders and ticks,
I see.
So then I went to this to work with spiders, and then the third professor, Igor Almeida, he is still at the University of Texas, El Paso. And he was from the same department, but he was already working with Chagas disease, and because my my interest was to work at, in these self-signal and post-translation modifications, so that's why, I went to work for Igor, because that was more like his line of research,
Mm-hmm. Totally. The
Yeah.
whole field of proteomics
Yes.
and so did you start working on mass spectrometry back then as well?
Back in São Paulo, yeah, as I was
Okay.
still undergrad.
Awesome. Awesome. So you've been doing this for quite a long time, specializing in this field, huh?
Yes, 20 years now.
That's incredible. So Ernesto, I mean, this is something also too that I admire about you and people like you, that you found some niche area that you really like, you really enjoy, and you stick with it no matter up or down or left and right. And is it because it's such a wonderful tool that lets you explore so many questions that we might have, and the better you get at using that tool, the more diverse type work that you could be able to do.
Yes, it's actually like one thing, drives you to the next thing. So
Yeah,
we usually do one experiment with proteomics. You have so many interesting targets that you find, and
yeah,
then you start to study one protein, and then that, calls for another protein, and you keep doing this and you keep going. And at one point, you start to have some idea of the whole pathway, but
yeah.
as much like we think that we know about all these signaling pathways, I still think that we only know the surface of it.
Right, right. So, I mean, this transition from Brazil to University of Texas in El Paso, this transition, I mean, must have been a little shocking to because of the language, but the culture,
Yeah.
as you said. What was it like? Because you guys were starting a new lab there.
Hmm. Well, I would give a step back, because I think my toughest transition was not that, was actually coming from the farm to São Paulo, right? Because
Okay.
your farm, your closest neighbor is a mile away, and then
Yes.
you suddenly now you're living in a city with 20 other million people.
Right.
You can barely, it's hard to go around because there's so much traffic, so many people. Culturally,
Yes.
it's very different too, and that I
Yes.
couldn't imagine that just 100 miles away, the culture would be so different, and it really took me quite a bit of time to adjust to São Paulo. No? So
Yeah.
when I did that, I thought, man, I can adapt anywhere. So
That's right.
going to Scotland was kind of a little shocking too, because I couldn't understand what they were saying because of their accent, I had English classes, but we're all American teachers, and
Yes.
and actually when I got to Texas was a little bit of relief, saying, "Oh, I understand something that people say here."
Oh, that's amazing.
Yeah.
That's awesome.
But actually, like it was quite when I got to El Paso, was quite a period of growth too, because the university was growing and still growing in such a fast pace, but there was a kind of the transition point. It started just a few years before, from an institution that was mainly undergrad institution to a research institution.
Mm.
So they got like investments, like they got the instruments, but they had not a lot of people to really who knew how to operate those.
Mhm.
So we got there. Actually, we were in an old building with the promise that the new building was going to be open in 3 or 4 months, and it took 2 years to be partially open. but it was quite the experience, because then they have like this big instrument there. Well, of course, it was not like the choice I would choose myself, but I had to use it because that was what we had. But
Right.
actually, we got a lot of data out of it. And then when we moved to the new building, I moved the whole instrument to the new building.
You did. Oh my gosh.
Yes, I only had help for people to help push it, but
Crazy.
yeah, but I actually went and unplugged it and plugged it in the new building.
Wow. Wow.
Yeah, and that was 2 years in my PhD, and then actually when we moved to new building, got the money to buy new instruments too. So it was a
Yeah.
good period.
Yeah. So, Ernesto, what would you say were elements that allowed you to take on that transition smoothly? You're away from home, different culture, different language. What did you do to help you get through homesickness, get through some of the emotional stuff that one goes through when they leave home and they arrive at a new place? And as you said, it's a moment of growth, and you realize that you're going through that growth. But what did you do to allow yourself to do that growth?
I think open mind, like you think that you need, learn a new culture, you're learning some new techniques, you're in a new place. I really did my best. I had colleagues that they were complaining about homesickness, that didn't like this there, didn't like that. But I think the key was to find things that you like to, right? So everywhere you go, you can find things that you like. You might have things that you don't like too, but try to find things that you would like.
Right.
The places that you like to go. Things that you like to eat. So that was I think the what helped me to move. To adapt and move faster.
Yeah. Well, and as you said, maybe the first shocking move to go from the farm to Sao Paulo and such a populous city, and not being able to get away from people even if you wanted to, type of thing. That must have helped you do yet that first leap of growth as well.
Yes.
um and so was there like, did you have to kind of teach yourself techniques for how to build good work habits, ethic, and like that kind of stuff? How did that?
So, some of these actually, I would tell you that my experience in the farm taught me the hard way.
Yeah.
Because
Yeah.
if you don't have work ethics in the farm, like you're just gonna fail yourself. Because like if you need to go and water the plants, and if you don't do that day, the plants are gonna die. It's period. Then you don't have harvest for the whole year, right?
That's
So if
right.
you don't go, you're lazy and you don't harvest that day. Next day, like they're overgrown. Then you have products that has a product that has no value. So there was a lot more. Let's say I had to have a lot more discipline there that would be required in academic setting. Because of course, like in an academic setting, oh, I was sick, I couldn't do my homework today. You can go talk to the professor. He will allow you to turn in the next day. I never heard about like the professor will not allow you to turn the next day, but like in a farm, it's not like forgiving like that.
Right, right. No, that is such a great point, and so true. I've interviewed a few animal scientists and farm related individuals, scientists now, that say, the farm makes you wake up early in the morning. You got to go milk those cows. You got to go
Yeah,
do stuff.
I used to wake up at like 4:45 in the morning to pack Brussels sprouts before I go to school.
Wow. Wow. That is awesome. That is awesome. So Ernesto, so the 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 world of proteomics and mass spectrometry, you kept pursuing that, and that's why we were excited, and still I remember our time with you so fondly here at Purdue, where you were really helping us lead the proteomics facility, and you've stuck with that as you've kept going, and now you're using it, I guess, more broadly to do other omics,
Yes.
lipidomics, metabolomics. um, I know that the techniques are quite different, but has that been something that you've taught yourself how to do, or have there been people along the way that you've relied on?
Yeah, it's actually both. Like when I was in grad school, my supervisor would like to analyze any kind of sample, so he would come and bring some collaboration. Here, I talked to this person at the meeting, and he gave these samples for you to run. I say, what are those? Oh, nothing, just some glycolipid. I say, glyco what? And then I would need to go and read how to prepare the samples and run, and it actually was very good training.
Yeah.
And then when I went to my postdoc, I was focused more on the proteomics and post-translational modifications, and that's the same at Purdue. But the difference is how we organize here at the national labs, right?
Mhm.
So you don't need to know everything. You just need to know people, actually.
Yeah.
So, and then what we do is like you have your project and you bring the people with the right expertise to your project.
Mm.
So, I often work with people that have expertise in metabolomics and lipidomics. I often like even I do quite a bit of post-translation modifications myself. Sometimes I bring people that they have some different knowledge than myself, so we work in as a group to we become more powerful, because now instead, like for me, say that I need to be running proteomics all the time and make asking my biological questions around proteomics, I can ask a little more broad questions now and just bring the right people to the team. Of course, that comes with the challenge now that you need to talk to people that might be from very different background than you. So
Yes.
yeah, so then I think that's why also that when I was at Purdue, I think it was a good fit to come and talk to the faculty, because now I had that background from my postdoc already, because we had always talked to people, even our mass spec group, we have people that are physicists, we have people around there, geochemists, we have what you can think of we have around,
Yeah.
and so we have very different backgrounds, so we need to make an effort to understand and talk in a common language,
Yeah.
yeah. Yeah, and but like what I really like about the at PNNL is, I'm not sure about if the other national labs are like this, but we we like to help each other. So
Mhm.
I never had anybody that here that I ask for help that will say, oh no, I can't help you.
Yeah.
If the person is not like the best to help me, he will say, oh, I'm not the best person, but this is the person you should talk to.
Right.
And it has been great.
Oh, that's wonderful. That's really good. And it's so good to hear, also a little bit the distinction between working in a national lab and working in academia. I'm certain that there are similarities and there are striking differences. Tell us a little bit about those.
So the similarities where we all do science, right? So we, as like any faculty at academia, we write projects. So
Mhm.
to the funding agents from to NIH to Department of Energy, Department of Defense, so all these different agencies, and when we get fund, then we work and do our projects. Also we have wet labs and all the equipment. There are a little bit the difference starts when how we are structured, in our personnel. So because we don't have students, the national labs, we don't have educational programs. We do have some educational programs,
Right.
but they're more like to advanced education for staff, or some cooperation with universities. But we don't have any program here that, to say that the master program or a PhD program. So our staff, they're very heavy, on postdocs and staff scientists. We do have interns from local universities. They come, in over the year, or we have interns that they come over the summer. So there are a lot of summer programs for people that are interested too.
Yep.
And but it's a lot more focus now on science, because we don't have that educational part. So we don't have a lot of these like dissertation committees, the classes.
Yeah.
So and the other difference is how we are structured. So we say that the people that work on my projects might be not the people that they are from my team, and which is kind of confusing. So we have they have the administrative structure, and then for each project we just build small teams for the project. So, and I can bring people from different places in the lab. It doesn't need to be from my team itself. So it's not like an academia that a professor gets a grant and then he hires a grad student, hires a postdoc, and these two people just work for his projects.
Right.
So we often share postdocs, even like the scientists, they're more like have this entrepreneurship, that they can work in projects they're interested on. And basically like they charge their hours through the project, and that's how it works.
Right.
So more on the administrative side, because I'm a scientist and also a team lead, so I do a little bit of administrative work, so in the administrative side is like how we organize. We are organizing directorate, so which is roughly how as the big schools are, say School of Engineering or School of Science, so like these big schools, and then under those the directorate. So we are under the Earth and Biological Science Directorate. We have the divisions. We have the biological science division, which one I'm in it. There is environmental and molecular science division, and there is atmospheric science division. So there are the three divisions of our directorate. And under this division, we have the big groups. So the division is about our division is about 200 people, and we have three groups of about 80 people each, 70 to 80 people each. So the groups are computational science, the biosystem science, and then the integrative omics, which I'm part on. So the integrative omics group is more a mass spectrometry group. So it's very heavy on mass spectrometry. We have inside them how many group teams we have now? I think it's 5 teams or 6 teams. I kind of get lost because we just changed the numbers. So my team is a biostructure and function. We have teams for proteomics. We have a teams for metabolomics, which also have lipidomics people. We have a team for instrument development and deployment. We have a team for more like mass spec operations, and we have a team for sample prepping. So we have actually 6 teams, and so each team has their own kind of strengths, right?
Yeah.
So my team, to say biostructure and function, we do a lot of mass spec too, and but we are probably the most diverse team in the group. And we have people that do cryo-EM, we have people that do NMR, we have, so the idea is now that we get all this, huge amount of data from proteomics and lipidomics and other omics. Now we're trying to, in my team, we're trying to find what is the mechanism, what is the function. So we work with the cryo-EM people, we work with the NMR people, we work with some of the microbiologists to try to really come up with the function of these molecules that we detect by mass spec.
Yeah, it's so neat. I can imagine that your rich background allows you to be a formidable partner and a great player in the teams that you are forming.
Mm.
Is there now, and you've probably been able to do this in such a way that you've participated enough and in teams that you've made also now this leap towards being a team lead. What does it mean to be a team leader? And how, like, was this a promotion that you obtained, or how does that happen?
Actually, it's a different path. So there is a path for like scientist path, and then you can go for all different levels of scientists. We have from 1 to 6, and we have also the pathways for limited term employees, like their postdocs, postbachelors, postmasters, and then there is the more administrative route. So then you start with the team leads. So the team, so the administrative organization, we have the teams first, and then that in here hierarchy it goes to the groups, division, and directorates.
Right.
So that's the first line of organization. It's kind of how it was a small department of the university.
Yep.
So the idea, what I do is to work on administrative stuff. If what people need is the paperwork for them to work in the lab. They
Right.
help them to work according to the government rules. And especially what I do is to help them develop their careers. So help they make connections, like strategize where they should put their time on, because to be a successful scientist, people might think, oh, you just need to publish papers, but that's not all, right? You want people to be a complete scientist, that they can go give talks and bring funding and reach out to people and participate in other projects that like just be able to do one single thing. So that's how the two pathways are different.
Yeah. Well, and I've known you, and I've known your style also, and your demeanor. You're a great people person. And I for me, it seems like a natural fit for you. But was there something that made you say, hey, I'm gonna maybe take this more administrative route rather than this other route? What was it that attracted you to that?
Yeah, so just clarify, I didn't give up the other route. So I worked 20% of my time in this administrative route, and they have like 80% of the time in this, it's still just really scientists, series. And then, the other thing that I think is a little bit different is that, that when we build the teams for each project, we don't really care about this hierarchy of, that if it's your team lead or your group lead, I work in projects from people who are under me on my team, and actually in my projects I have my statistician is actually the division director, who is two steps above me. So we just have fun with this. It's not like an academia that is very hierarchical, right? So all the students call like professor, doctor. Here, like if anybody calls me a doctor, saying, who is this person talking to?
Right. Yeah. No, that's so great to hear a little bit about what the culture is and what the atmosphere. And it was just mainly, a question, because I'm sure there are people that are listening that say, oh, I didn't know that this was an opportunity that I could take advantage of, because there are people that might find that attractive, or might say, oh, I didn't realize that that also was something that I could entertain as an option.
Yeah, it is. And going back to your question, why pursue this pathway? I'm not sure if you heard about, but my son had retinoblastoma.
Mm-hmm.
So and there was like quite a bit of you know quite a bit of weight, having to travel from here to Seattle, to be treated in Seattle. So
Mm-hmm.
Seattle is 3.5 hours from here. We had to go every couple weeks and spend 3 or 4 days there, they come back.
Yeah.
And then during this time, the group, the team and the group, they put a lot of effort to, help me, to cover the meetings I used to lead. They had the meetings by themselves. They organized by themselves. They
Yeah.
look for the deliverables by themselves. They, so when everything was done, and thanks God it's all done because my son is doing pretty well.
Yes.
And I thought I should do something to retribute to the group, and like become the team lead was a way that I thought I could, return these, all this that he gave me,
Yes.
and could have some impact.
Absolutely. What a wonderful gesture. And again, like, how awesome to hear that when you needed them, they were there for you and they backed you up, and
Yeah.
in the time of need. And it sounds again like a great culture, great atmosphere, where people are able to live life and work with colleagues that are caring and that are also there for you. It sounds like a wonderful wonderful atmosphere and environment. Ernesto, what is like, is there something that you would want people to know before they start going towards an opportunity, a job opportunity in a national lab like PNNL? What should they do to get ready, to prepare?
I think the first thing is have open mind. We all were from somewhere, right? That people might think, oh, I'm not qualified enough. Like, you just give us the problem to evaluate. If you cannot be valuable, but you know, a lot of people don't apply because they not think they're qualified enough, but actually
Mm,
they're really well qualified.
yeah.
And the second thing is that we do a lot of training. So we're not expecting that you come and you're just gonna be doing that thing forever. We do have quite a bit of training. We train people. Even like my first year as a postdoc here was mostly like very heavy on training because the culture, the operations are quite different. So
Mm,
we are very really careful on safety aspects. So if somebody's doing something that we think is not safe in the lab, we can just go and tell the person, oh, wait, stop.
yeah.
I think that you're independently if it was not like right or was right or not right, we go and we tell the person, stop. Let's reevaluate.
Mhm.
Let's ask the biosafety people if that is the right way to do. The other thing also to consider, what I like is the environment is very cooperative because one thing that makes them more cooperative because we need to work in different projects, right? So if we can't cooperate with people, we cannot survive here. Right?
Yeah.
So even like myself, right? So I write a proposal in IH, and IH will say, oh yeah, I will fund you, but we don't want your time in this project to be more than 25% because you're PI, you're very expensive,
Right.
and you're gonna chew all the budget, just for you.
Right.
So then I need to go and work somebody else's project too to help cover the rest of my time. So then that make the environment to be more cooperative, like so people are really always willing to help, because that's the way that we can, do better science. We can, stay employed, and we can come up with the next proposal that are even better.
Yeah, absolutely.
And another thing that people should consider too is that we value a lot the balance between work and life. So we are not expecting people to come here work 70 hours a week. Of course, that 40 hours might not be enough. So actually, people, they might work a little bit extra, but we want that the people to come, when they come, be very productive, but they have their time to to rest, to have their their hobbies, because we know that if you work under stress and pressure too much, and I'd say that not that to tell the I think it needs to have a distinction between, say, people think that we're lazy or not. We want the people to really come in to do the best.
Yeah.
So we know that that you work the 7 hours a week, you're less productive actually than working the right hours and take your time to recover and everything. So we are really prone to like the productivity, and people can see from even my publications now, I'm very productive in my field. So that is how we operate. And even like when the person comes as a postdoc, they have all the different ideas. Well, calm down. You have all these different ideas, but which one is like that thinking your time is the most valuable thing, asset that you have in your life.
Yeah.
Is your time. Where are you gonna invest your time? Now that is like the key question that you need to ask every morning.
Yes.
Your time is the most important thing in your life, the most expensive thing in your life. Where are you going to spend it? Where are you going to invest it? Because we can't do everything, but we should be planning and working, and really have a good plan to, when we do experiment, to get the when you get the results that need to be conclusive, then let's do this just to see what happened.
Yeah, no, and I think that that piece of guidance is so important for people to realize that their time is their biggest asset. That's something you can't throw away, and yet a lot of people do throw it away. A lot of people think
Yes.
that, oh, it's so I could do that later and procrastinate, or I'll just do a little bit of this and then, not because I have something else, some other distraction that is taking. And I mean, think about our cell phones. Our cell phones are constant time consuming distractions that are with us all the time.
Yes.
They don't give anything back to us. They keep consuming our time and consuming our energy. But it's such a great piece of advice that really wake up every morning and prioritize what you are going to do with your time. Because if you don't, then time runs away from you, and hours become days and days become weeks and months, and it's very interesting how we get that productivity. And I think maybe even tying back to what you said before, the work ethic, having those good habits of building on what you've already planted, meaning like if you're a farmer, and as you said, like if you don't work every day on what you planted, come harvest time, you're not going to have much to harvest. And life is like that. It takes a lot of time, and it takes a lot of investment every day to make sure that at the end, when it's harvest time, you're getting a good harvest out of your investment, your time that you put in to make sure that whatever you planted it grows, and it grows in a robust way. I really like this conversation, Ernesto, and I think, you know, it's so natural to talk to you and to catch up in this way. Is there anything that I didn't ask you that you wish I would have asked you?
No, I mean, I think the other thing that I had to say for people that might be interested on the national lab career is that we we give a lot of training, so that's something that they should have in their mind when they come here, when they go to their next job, right? So you don't want like a job that just want you to do the same thing you already know how to do over and over again, because that is like you become like a more like a assembly line employee, right?
Yes.
And the other thing that you should consider, like even like for the students, if want they come to the postdocs, our postdocs are well paid as well. So we pay. It depends a bit on the career and the expertise and the experience, but we usually pay much better than the academia postdocs too. So there's something else to consider as well.
Absolutely. And it's so good. And if somebody wanted to look into what opportunities are available, let's say in your group, could they contact you through LinkedIn? Could they
Oh, yes. Either LinkedIn, our emails are easy, so it's my first name dot last name at pnnl.gov. So everybody can find that.
Perfect. Or even, I guess, on the PNNL website, they could find what opportunities are open, what are the different groups, and so on.
Yes, yeah. It's we don't have very, let's say, good, I think, websites for like the different projects and things, because things have changed so fast and it's hard to keep up. But the other opportunities on their website for job opportunities they're always full of job openings there.
Okay, well, great. Thank you so much. I really appreciate this time with you. Dr. Ernesto Nakayasu, the Biostructure and Function Team Lead at the Pacific Northwest National Lab. Thank you so much for talking to us and being our Pathfinder today.
Well, thanks so much. The pleasure was mine.
Well, I hope you have a great day. Good to see you too. Bye-bye.
See you. Bye.
Thank you for joining us once again, and remember to hit the like button and subscribe to our channel.